Hello and welcome to the next class of embryology, and today we shall be discussing about important aspects of genetics and molecular biology. In case of genetics and molecular biology, our aim is to understand in case of a human, how does this topic apply to the human embryology, because yesterday I told you that how important the cell division is. Yesterday I was telling you that how important the matter of how important is the division of cells. All right, I think am I audible or not? Yes. Hello. 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 All right. Give me a few seconds to settle down. I'm just checking the audio and video quality. I think video quality is okay, but I'm not finding any audio. Yeah, audio is fine. Yes. So let us begin our discussion. Not wasting much time, I want to talk about genetics first. In our classes in embryology, first I'll discuss what are the important aspects we will be discussing today. I'll summarize what we'll be discussing today, and after that I'll discuss the topic proper. So today we shall be discussing what is genetics all about. Today we shall be discussing genetics, that genetics is a branch of biology which deals with the study of genes, inherited characters that are acquired from the parents, that go from the parents to the offsprings at the time of fertilization. Remember, all this transfer of genetic material that occurs at the time of fertilization, and the cell which is carrying it, all the lakhs of characters, I'm not millions of characters that a human being has, are carried by a small cell, one each from the parent, which fuses to form a zygote, and in the zygote the division occurs. We'll be dealing with all that today. We'll be talking about, so this is the first thing that we'll be dealing. We'll be talking about how are these genes connected to the strands in the DNA. What is DNA, what is RNA, we'll be discussing very briefly. In detail, we'll be discussing this topic in biochemistry, but here I'll be discussing what is pertaining to the embryology topic. Next, we'll discuss what are the number of chromosomes. We have got, each gamete has 23 chromosomes. these two gametes will fuse to form a zygote, which has got a 23 pairs of chromosomes. 23 pair means a diploid number is 46. Diploid number is 46. Out of that 46 chromosomes, we have got 44 chromosomes are called as autosomes, which carry vegetative characters, and 2 chromosomes, they are called as allosomes or sex chromosomes. All right, sex chromosomes, and they determine whether this zygote will become a male or a female. So if it is 46, we call it 46. If it is XX or XY, so XX we are calling it as a female. This is all biology. We already you might have read it in your earlier schooling days or college days. And if it is XY, the zygote is a male zygote. All right, this will be discussing. Then we are summarizing what we'll be discussing in today's class. So today we also discuss how the cell division is taking place. We'll be talking about the mitosis phases, the mitosis and the meiosis, the two different types of cell divisions. We'll talk about the growth factors responsible. We'll talk about the different diseases that can occur in case of inheritance, like X inheritance or Y inheritance, diseases related to the X chromosome, diseases related to the Y chromosome we'll be discussing. And in the end, we will be discussing development, differentiation, and the growth changes that are brought about by these genes. Please remember that genes are producing certain factors. We are calling it as transcription factors. We'll discuss how the transcription is getting controlled by the RNA transcription, protein assembly, and protein manufacture. We'll discuss all that. All right, so this is all about today's the summary of today's class that we are going to discuss. So let me start the topic. I'll talk about genetics first. So what is genetics? Genetics is a branch of biology. Genetics is a branch of biology that deals with transmission of inherited characters. It deals with the transmission of inherited features or inherited characters or inherited traits, we call it. So color of our skin, the type of our hair, the voice, the face, the various other features, physical features, psychological features are from our parents. All right, and the study of all these characters, few are dominant. If they are expressed, we call it dominant. If they are not expressed, you call it recessive. We call it recessive. All right, so this is how we'll deal with in the study called as genetics. Now please remember, the inheritance of these characters is brought about by these dominant or recessive genes. All right, so genetics is actually study of genes. You can call it genetics as study of genes. All right, and the mode of transmission that occurs, so the hereditary factors and their expression during development, that is prenatal life, the postnatal life, the embryonic life, that is prenatal, and postnatal life, which we already discussed the definitions yesterday. So we'll discuss, that is all discussed in genetics. Now you may wonder, sir, what is gene? Please remember, gene is made up of nucleic acid. Genes are made up of nucleic acids. What are the nucleic acid? We are talking about deoxyribonucleic nucleic acid, so that is the DNA. The nucleus of a cell is made up of this DNA, deoxyribonucleic acid, and it contains chromosomes. That is chromosomes. The DNA known as chromosomes. The genes are structured in the DNA. We are calling it as chromosomes. I told you that there are 46 chromosomes, 23 from each parent, which fuse to form, which get paired to form a zygote. Now these chromosomes in our entire life, they code for enzymes, hormones, antibodies, characters, proteins, and various other features which are essential for bringing about a normal life till you die. All right. Genes exert important influence on cellular function. So right at the level of cell, the molecular level, the genes are acting to synthesize proteins, and these proteins are formed by the collection of amino acids. So these proteins determine the eye color, the hair color, the type of hair, the face color, the face type, the skin color, everything. All right. It also it also helps us to analyze and characterize the things that you like, things that you dislike, the way you behave. All right. Few things obviously are learned as you grow, but there are certain inherent tendencies that which change our personality. So let us now talk about DNA. The basic structure of DNA we'll discuss in our biochemistry class. I'll be discussing all that. I'll be discussing about Watson and Crick model of DNA, the double helix model of DNA. I'll be discussing in detail the double helix model of DNA. I'll be discussing various bonds and how this nucleic acid is formed in that class. But here what you need to know is, quickly we'll try to revise that there are two strands in the DNA. Two strands are linked to each other at regular intervals. So say suppose this is one strand, this is another strand. We are calling this model as a double helix model. They are linked together with the help of certain bonds. We'll discuss what are these bonds later on. So two strands which make just give me a second. All right. So at regular intervals. So each nucleotide, please remember, DNA consists of a chain of nucleotides. So these are nothing but, this strand is nothing but, it is containing a chain of nucleotides like this. Nucleotide. So we are calling it as nucleotide. What does each nucleotide made up of? Each nucleotide is made up of a deoxyribose sugar, a phosphate, and a base. It is made up of deoxyribose sugar plus a phosphate bond plus a base. This forms a nucleotide. This forms a nucleotide. So each of these strands is made up of so many nucleotides. Now you may wonder, sir, what is this base? This base is either adenosine or adenine, or guanine, or cytosine, or thymine. All right. So please remember that these two strands are complementary to each other. That is, the adenine always pairs with thymine, guanine always pairs with cytosine. This is called as complementary to each other. The property is called as complementary to each other. That is, the linkage is peculiar in one strand. If at all we have got, say suppose A here, so opposite side is always T. If you got, say suppose guanine here, opposite side is always C. So this is about DNA. In case of RNA, the C is replaced by uracil. So in case of RNA, C is not present. It is replaced by uracil. So in case of DNA, it is adenine, thymine, guanine, and cytosine. And the two strands are complementary to each other. You call it complementary strands. The bases complement each other. What are the bases? I told you, A, T, G, C. The short forms are A, T, G, C. A, C and T, like that. All right. So this is all about the ribonucleic acid, the deoxyribose. If it is a deoxyribose sugar, so this ribose is a sugar. Then we have got a phosphate bond, and then we have got a nucleoside base. All right. The ribonucleic acid, the deoxyribonucleic acid, the structure of if you talk about RNA, it is ribose sugar. All right. RNA is a ribose sugar, and instead of the base thiamine, it contains uracil. So instead of thiamine is not present. In case of thiamine is replaced by uracil. So it is thiamine which is replacing. There are three main forms of RNA. We have got mRNA, we have got tRNA, and we have got rRNA. All right, mRNA, tRNA, and rRNA. We have other forms of RNA which are having certain different function, like we have got microRNAs called as miRNAs and small interference RNA called as siRNAs. We'll discuss this in case of microbiology. Microbiology and also biochemistry, right? So please remember that the components that are required for expression of a gene, we need certain growth factors. We need growth factors for expression of gene. Then we need receptors so that they recognize these growth factors. Then we need signal transduction factors or signal transforming factors or signal transducing proteins, which convert the signal from the cell membrane to and bring about activity in this cytoplasm. And then in the end, we need activation of transcription factors. Activation of transcription factors. What are these? These are the components of gene expression. If they ask you, what are the components of gene expression, then you should answer, these are the components of gene expression. So if there is a cell, we need a growth factor, we need a receptor on which this growth factor sits and acts. Then we need the signal transducing proteins. Then on the DNA, we need, on the DNA or RNA, on the nucleus, we need the transcription factors which need to be activated. These transcription factors bind to the DNA of the nucleus and finally produces a transcription or gene protein. Because of this transcription, what happens is the desired protein or enzyme is formed, and thus the activity is taken care of. So what are the growth factors and what are the differentiating factors? For example, I'll give you examples of growth factors now, which are very important for molecular biology level. All right. So first important growth factor we'll talk about is epidermal growth factor. The epidermal growth factor is responsible for growth and proliferation of ectodermal and mesodermal components. So short form is epidermal growth factor, EGF. Then we have got the transforming growth factors. Transforming growth factors. The transforming growth factors are named by the letter beta. We have got beta 1, beta 2, beta 3, beta 4 and beta 5. They are responsible for formation of epithelial branching. They are responsible for myoblasts, growth of muscles, myoblasts. They are responsible for epidermal branching. They are responsible for epithelial branching. They are responsible for formation of extracellular matrix. These are the roles of transformation or transforming growth factor beta. Next, we have got bone morphogenetic factors or bone morphogenetic proteins. Bone morphogenetic factors or bone morphogenetic proteins. The name itself indicates that they are responsible for bone formation. They are responsible for cell division. They are responsible for migration, apoptosis. Next, for example, we have got Mullerian inhibiting factor. This is a very, very important growth factor. If this growth factor is absent, then the genitalia of the fetus will not grow properly. All right. Mullerian inhibitor factor. Then the name itself suggests that it is suppressing something. So what does it suppress? It suppresses the paramesonephric duct. This paramesonephric duct is responsible for what? For growth of the uterus and the vagina. That is the female genitalia. So Mullerian inhibitor factor, if it is present, it results in growth of the male genitalia by suppressing the female genitalia. And if it is absent, that results in formation of the female genitalia. All right, Mullerian inhibiting factor is very, very important. We'll discuss different types of genetic abnormalities that result in ambiguous genitalia. All right. Then we have got certain different fibroblast growth factor, insulin-like growth factors. I already discussed insulin-like growth factors responsible for fetal growth, nerve growth factors and all. The name itself indicates. So we are discussing all those factors. We have discussed important factors in this session. Next, let us talk about the chromosomes. Single set of chromosome is called as haploid chromosome. Two sets of chromosomes we are calling it as a diploid chromosome. If it is single, you are calling it as a haploid. Haploid set is termed as N. And if it is paired, double, you are calling it as a diploid chromosome. The number of chromosomes in each cell is fixed in every species. In human beings, it is 46. 46. I already told you this point. One set of chromosome is haploid. The two sets of chromosome, each from each parent, you calling it as diploid. We have got 44 autosomes and two sex chromosomes, which can be 2X. Both the sex chromosomes may be X in a female, and then 1X and 1Y, which determine a male. Then we'll talk about typically what are the parts of a chromosome. So please remember that chromosome has got a short arm and a long arm, and the tips we are calling as a satellite. So we have got a short arm like this. We have got two short arms in a diploid chromosome, and the centermost portion where these short arms and go and attach, it's a centromere. We have got two long arms of a chromosome, spindle shaped arms like this. So we have got a tip of the short arm. We have got satellite. This part is called a satellite, which communicates with other chromosomes. So we have got, so this area is a constriction, is it not? So this is a primary constriction, and this area is called a secondary constriction. So we have got short arm here, designated as P. P stands for petite. And we have got long arm of a chromosome. Long arm of a chromosome. And in the center we have got centromere. So these are the portions of a chromosome. So quickly we'll try to understand what is the significance of chromosome. Significance of chromosome is this is carrying a DNA sets of genes, and a fully formed chromosome consists of completely paired strands complementary to each other, and they have got a wonderful arrangement of these genes which are responsible for the expression of a character. Study of chromosomes is called as the study of the mapping of chromosomes is called as karyotyping. So if I do your karyotyping, I'll come to know whether your chromosome is normal or not. We call it karyos. Karyos means chromosome, typing means mapping. So mapping of a chromosome, the study is called as karyotyping. This can be done on clinical basis. If you are suspecting some chromosomal disorder in a patient, you can send the patient's blood sample for karyotyping. All right. So we classify chromosomes based on this centromere. We classify it into metacenter, submetacenter, acrocenter, telocenter, and all. That is not important for embryology. That is not important for your at this level. All right. What is important at this level is any structural abnormality of chromosome which may result in certain disorders. What are the structural abnormalities of chromosomes? So the abnormalities of chromosomes include, first is deletion. So deletion, the name itself is indicating that there is loss of some segment of chromosome. Loss of a segment of chromosome. For example, in a syndrome called as Cry-Du-Chat syndrome. There is a 5p deletion. So we have got 23 pairs of chromosome, is it not? In this 23 pairs of chromosome, we call it first pair, second pair, third pair, fourth pair. It has got first pair short arm, you are calling it as a 1p. First pair long arm, you are calling it as a 1q. Similarly, we have 2p, 2q, 3p, 3q, 4p, 4q, 5p, 5q, something like that. So 5p is Cry-Du-Chat syndrome. Deletion of 5p, you are calling it as a Cry-Du-Chat syndrome. Cry-Du-Chat syndrome. Sometimes if it is partial deletion, you are calling it as a micro deletion. This is another example of abnormalities of chromosome. The second example is deletion which is detected by high resolution testing. You detect it. For example, if the proximal part of long arm of chromosome is called as what? Q, is it not? So 15q, 15th chromosome, long arm. If the proximal portion or small portion of long arm is deleted, it results in either Angelman syndrome or Prader-Willi syndrome, depending on the inheritance. If it is paternally inherited, that is coming from the father, it results in Prader-Willi syndrome. Prader-Willi. And if it is maternally inherited, it results in Angelman syndrome. What are the features of these syndromes? It is not important now, but in medicine, I'll definitely, when I am teaching medicine genetics, so I'll discuss the important clinical features of this Prader-Willi syndrome and Angelman syndrome. In case of Prader-Willi syndrome, the macro testes and various other abnormalities. In case of Angelman syndrome. Now, please remember that mother is angel, is it not? Your mother is an angel. So we consider mother as angel. Just to remember that Angelman syndrome is from the maternal inheritance. Next important abnormality that we'll discuss is inversion. Inversion is an abnormality that results from detachment of chromosomes, detachment of a part of chromosomes. All right, and this inversion, it does not cause any clinical syndrome. Definitely it's a problematic thing, but it will not cause, it is not expressed as a clinical disorder. Next is we have got duplication of chromosome. Sometimes what happens, the chromosome, there is abnormal splitting of chromosome results in duplication. All right, twinning or duplication. The most important abnormality that comes into our practice is translocation. What is translocation? Translocation is exchange of segments between the non-homologous chromosomes. For example, between the non-homologous chromosomes, for example, we have got a set of chromosomes. Say suppose this is a set of chromosome number 9 and this is a set of chromosome number 22. So 9 to 22 translocation, this may result in Philadelphia chromosome, we call it. This may result in the leukemias. 9-22 translocation. So what happened here? Exchange of segments between the non-homologous chromosomes. From 9th chromosome to 22nd chromosome, there was an exchange of genetic material. We call it translocation of genes. From 9th chromosome to 22nd chromosomes, few genes get transferred, and that results in a disorder, and that is called as that is called as translocation. So duplication or multiplication of chromosomes will also result in certain disorder. For example, sometimes numerical abnormalities. Please remember that trisomy 21. See, please remember we should have a set of chromosomes. So we have got first pair like this, something like this, second pair like this. So like this we have got 23 pairs of chromosomes. Sometimes what happens at 21st chromosome, at the location of 21st chromosome, there is presence of third chromosome. We call it trisomy 21. Trisomy 21, please remember, it is Down syndrome. Very famous genetic disorder. You might have seen it has got quite high incidence, that is resulting from delayed age at pregnancy. Mother's age is very high, more than 35 years at pregnancy. There is very high chance of this chromosomal abnormality called as trisomy 21. You can detect this trisomy 21 by antenatal tests, so that you prevent this birth of a baby which is having lifelong mental retardation, right? So sometimes trisomy 18, at the 18th position, if you have an extra chromosome at 18th position, you are calling it as a trisomy 18. This is Edwards syndrome. Sometimes at 13th position we have got trisomy 13. So this is called as Pettau syndrome. Cleft palate, chromosomal abnormalities resulting in cleft palate and ventricular septal defects. That is a congenital heart disease. It's a feature of Pettau syndrome. Edwards syndrome results from trisomy 18, which has got congenital heart disease. Then sometimes what happens, sex chromosome, absence of sex chromosomes, that is 45 or XO. Instead of 45 double X, we have got absence of chromosomes. So you are calling it as 45 XO. This is Turner's syndrome. Turner's syndrome. Next we have got 47 double XY. This 47 double XY is one extra chromosome, one extra X chromosome has come. We call it Klinefelter syndrome. This is how the sex chromosomal abnormalities and the autosomal chromosome, that is vegetative chromosomal abnormalities, have been mentioned. So these are all the abnormalities of the chromosome. All right. So if I say that the chromosomal karyotyping of a person is 45XX, what do you suggest? I call it as a female, normal female, is it not? Normal female karyotype. If it is 46XY, I call it as a male karyotype. All right. And if I say that 46 by 47, if I say like this, I'm talking about mosaicism. All right. What is mosaicism? I'll discuss later. Next, if I say it is DEL, small d, if I say it is small d, if I mention it as DEL, I mean that deletion. If I say INV, I mean that there is inversion. So these are the denoting formats, short forms or symbols of karyotyping analysis. If I say small r, I'm calling it as a ring chromosome. Ring chromosome. If I say DUP, small d, small u and small p, I'm calling it as duplication of chromosome. So this is how karyotyping abnormalities are mentioned. In exams, sometimes you will have to write what is known as pedigree chart. We'll try to analyze what is pedigree chart. This is our next topic of discussion today. So for a patient, you need to draw a pedigree chart. To understand the pedigree chart, please remember it's a pictorial representation of generations of a family. Showing the information of all the family members. So if you want to do pictorial representation showing all the family members, if you want to show all the family members details, whether the mother and father have been consanguineous, that is related to each other, blood relation, consanguinity, live birth, stillbirth, deceased birth. So we discuss it like this. There are certain denotions of pedigree charting. Pictorial representation is called as pedigree charting. It is very helpful in case of genetic diseases. For example, if I draw a small square like this, it is a normal male. Circle like this denotes a normal female. All right. If I draw shaded, say suppose if I draw a shaded, square like this. A shaded square denotes what? Affected male. It means affected by some genetic disorder. If I draw a shaded female, shaded circle like this, so obviously it's a affected female. So you should understand all these points. You should understand all these points of pictorial representation of pedigree charting. Then if I say that, if I draw a small rhomboid like this, that is sex is, we do not know what is the sex of this person, unspecified. All right. If I draw a small circle, small dark circle like this, this is denoted as abortion. If I draw a square and if I cross it like this, it means the person is dead. All right. And if I draw a small circle with a central dot, it means this person is suffering from a X-linked recessive carrier. The person is carrier. X-linked recessive carrier. All right. And if I draw a square or a circle which is half shaded, it means I am dealing with a heterozygous character. Either male or a female. All right, so these are all the denoted formats of pedigree chart. In the pedigree chart, we also have something like this. If a square and a circle, if you're drawing a single line, it means they are mating with each other to produce offspring. So parents, we can call it mother and father. If I draw two lines like this, it means they are related to each other, consanguineous, blood relatives who got married. All right. And then if I draw a flow chart like this, either squares or circles, it means it is offspring. Offspring means children. If I draw a diagonal flow chart like this, a diagonal flow chart like this, and if I draw characters like this, all right, it means they are twins. Obviously here in this case we have got dizygotic twin. I'll tell you what is dizygotic and monozygotic twin when I'll be teaching gynecology. Then if I draw like this, and if I draw characters here, and if I draw a line in between these two, then I'm talking about monozygotic twin. Then I'm talking about a monozygotic twin. All right. At any point of time, if on a square or a circle, if I mention like this, it means I'm talking about the index case. Index case means the patient. I'm talking about the person whose pedigree chart I have drawn. Now I'll draw a pedigree chart and I'll show you one example. So circle means female, square means male, they married to each other. After mating, say suppose they have got 4 children, and say suppose they have got, first child was female child, second child was also female child, third child also female child, and then the fourth child was a male child, for example, just an example. And then again the first female child got married to a person, and she again has 3 children. 2 are females and 1 is male. And you are talking about the third generation, this female, your patient is this person. And in this family you find out that they are carrying a gene, that is mother is having a heterozygous trait for some character you're talking about. Say suppose brown hair or color of eyes, or it may be n number of things, like we are talking about hypertension, diabetes or something like that. All right. And say suppose this person you're talking about is suffering from some genetic disorder. So she is an affected female. All right. So you shade it like this, affected female, something like this. So this is a pedigree chart, we call it. Pedigree charts are important for USMLE exams. Pedigree chart are important for certain exams, like if you're going for higher specialty exams, subspecialty exams, then obviously pedigree charts are important. So we'll discuss pedigree chart based problems separately when we'll be discussing pathology, genetics in pathology. All right. So we should also discuss now what are the autosomal dominant traits and what are the autosomal recessive traits. Autosomal dominance means 50% of the offsprings develop the disease. All right, 50%. For example, say suppose in this chart which is in front of you, say suppose 4 children of this first gen. So this is first generation, this is second generation, and this is third generation. Okay. If you want to say that in this pedigree chart we have got autosomal dominant disease, so 50% of the offspring should be suffering from the disease. So 50% of the offspring means in the second generation 2 children. All right. Then in the third generation also again at least 50%. That is 2 children. are suffering from this disease. So this green color is denoting what? It is denoting the autosomal dominant trait. So autosomal dominant means what? A disease or a disorder or a trait which is seen in at least 50% of the offsprings in every generation. It is not exceeding that, or it is not less than that. Both male and females are equally affected. That is most important point about autosomal dominant. So both male and female are equally affected. Alright, examples for the autosomal dominant trait are hereditary angioneuritic edema, then osteogenesis imperfecta, then we have got HOCM. HOCM stands for hypertrophied obstructive cardiomyopathy. HOCM, hypertrophied obstructive cardiomyopathy. Huntington's chorea. These are all the various disorders which present as autosomal dominant trait. We call it AD, short form. Then we talk about autosomal recessive inheritance. Autosomal recessive inheritance denoted as AR. Autosomal recessive inheritance means that there is a history of consanguineous marriage in this person's parents. Parents are both blood related and both of them are carrying an abnormal gene. 25% chance that this gene is affecting the offspring. There is a for example, I'll draw a trait now. So male, consanguineous to female. They have got 4 children, irrespective of male or female. They have got 4 children, so suppose. I'm just giving you an example. You can write multiple types of examples here. So only one person, either male or female, is affected. You are calling it as. So here, what is the offspring affected? 25% of the offspring is affected, is it not? So you are calling it as. You are calling it as autosomal recessive. Autosomal recessive, both male and female have equal chance. The examples of autosomal recessive inheritance are cystic fibrosis, sickle cell anemia, thalassemias. There are many examples we'll discuss in pathology. For example, you remember all the inborn errors of metabolism are autosomal recessive. Or all the enzyme deficiencies are autosomal recessive. Please remember this one. Similarly, we have got third type of inheritance disorders called as X-linked diseases. X-linked inheritance. Remember, in a female we have got 2 X chromosomes. In a male we have got single X chromosome. In X-linked inheritance, the rule is males are always affected. Why? Because they have single X, and males always affected. This is the first rule. Second is 50% of the females are affected. 50%, all the females are carriers. All the daughters are carriers. All the daughters are carriers. It may affect. So daughters are carriers here. It may affect female, and if at all it affects, there is a 50% chance. The examples here are X-linked disorder. Example is X-linked vitamin D disorder, that is X-linked rickets we call it. So example here for X-linked inheritance is X-linked rickets. There are many examples I'll discuss when I'll be discussing pathology. In X-linked inheritance, we have got one more inheritance called as X-linked recessive inheritance, where all the females are carriers and all the males are affected, and this disorder is hemophilia, X-linked recessive. Example is hemophilia. So here daughters are carriers, they're never affected, and sons are always affected. Sons are affected by the disease. There are certain royal families of London who are suffering from this disorder. So very, very famous X-linked recessive disorder because of consanguinous again. For example, color blindness, hemophilia, color blindness, then Duchenne muscular dystrophies, which are X-linked recessive. Similarly, we have got Y-linked inheritance also. Y-linked inheritance, obviously it is seen only in males. Inheritance seen only in males, and this is affecting. We are calling Y-linked inheritance as Holandric inheritance. We are calling it as Holandric inheritance. Holandric genes that carry Y-linked traits, we are calling it as Holandric genes. For example, hairy pinna, that is hair on your ears. If you can see this trait, it is commonly seen in males only, not in females. So you are calling it as Holandric inheritance. So we have got autosomal inheritance and X-linked inheritance and Y-linked inheritance or sex-linked inheritance. So these are all the different disorders. With this, we complete the understanding of chromosomes. All right. With this, we complete the understanding of chromosomes. Now, time has come. We need to understand the importance of cell division. To understand the importance of cell division, I have few diagrams for you so that it's very easy for us to analyze these diagrams. I'll just post these diagrams. So cell division is divided into mitosis phase and meiosis phase. If you can see this diagram, cell division is divided into mitosis phase and meiosis phase. Mitosis and meiosis. So we have got mitosis and meiosis. Mitosis is division of cells into two daughter cells. Alright. Mitosis is the daughter cells, in turn they have their own activity and they are ready for another division. Alright. So first of all, you need to understand that a different kind of cell division is occurring in the background process. We are calling it as meiosis. And meiosis is, it is occurring at the formation of sex chromosomes, sex cells, that is gametes. Please remember that meiosis consists of two successive divisions, meiosis 1 and meiosis 2. Meiosis 1 and meiosis 2. So division of all the vegetative cells or somatic cells, we are calling it as mitosis. Division of gametes, we are calling it as meiosis. Meiosis is again divided into meiosis 1 and meiosis 2. Meiosis 1, that is the first phase of meiosis, and meiosis 2, the second phase of meiosis. The cells resulting from meiosis, that is the gametes, they differ from adult cells in the fact that the number of chromosomes is reduced to half the number. The number of chromosomes that is reduced to half the number. This is first important point. The genetic information of various gametes is not identical. For example, if you can follow this diagram, if this diagram is yes, so we are talking about meiosis 1 here. So in case of meiosis 1, the number of chromosomes is reduced to half. the number of chromosomes is reduced to half the normal number. For example, the division has started with chromosome number 4 chromosomes here, if you can see 4 chromosomes here. After prophase 1, metaphase 1, anaphase 1 and telophase 1, the number has reduced to half. This is meiosis. Meiosis means division of chromosomes. Division of chromosomes. This is meiosis. Meiosis is division of chromosomes. After that, each cell undergoes meiosis 2. Alright, in case of meiosis 2, each cell will receive one set of chromosomes. So initially there were 4, from 4 it has become 2, from 2 it has become 1. So this is meiosis 1 and meiosis 2. What are the different phases of mitosis and what are the different phases of meiosis? Alright, let us try to understand. So in case of meiosis, the mother cell is undergoing, in case of mitosis, the mother cell is undergoing DNA replication first. So initially the DNA divides, then the cell division occurs and 2 daughter cells are formed. Both the daughter cells have, same as mother cell, they have got 2 chromosomes each. This is mitosis. But in case of meiosis, each daughter chromosomes will have half the number of chromosomes of mother. For example, mother here has, the parent cell has got 1, 2, 3, 4, if you can see. So 1, this is 1, this is 2, this is 3 and this is 4 chromosomes here. And in the end, after meiosis 1, if you see, the daughter cells have reduced to 2 each, 2 chromosomes each. Alright, 2 chromosomes each like this, 2 chromosomes each, the 4 daughter cells. Then after meiosis 1, again each will have one single set of chromosome. This is meiosis 2. What are the important phases of mitosis and what are the important phases of meiosis? What are the important phases of, we have got prophase, metaphase, anaphase, telophase. We have got prophase, metaphase, anaphase and telophase. Prophase, metaphase, anaphase, telophase. What's happening in the prophase? During prophase, the chromatin of the chromosomes becomes gradually more and more coiled so that the chromosome becomes recognizable as a thread. Chromosome becomes recognizable as a thread-like structure and later on it acquires a rod-shaped structure. Rod-shaped structure. Towards the end of the prophase, the two chromatids constituting of chromosome become distinct and the changes that are occurring in the chromosomes are very much illustrated in case of prophase, very much enhanced in case of prophase. Here what's happening is microtubules. If you can see this diagram, I'll draw with a small color like this. So these are the microtubules. So if you see, microtubules are formed which holding the chromosomes, segregating the chromosomes. So we have got microtubule assembly. There are certain anti-cancer drugs which will kill this microtubules assembly and thus they prevent cancer, division of cells in the cancer phase. Alright. Next comes the metaphase. Metaphase is formation of spindles. So metaphase is characterized by spindles, spindle formation. The metaphase, the chromosome moves in a position in the midway. Meta means middle. Alright. So if you can see, chromosomes have moved here. So chromosomes have moved here in the diagram. Is it not? The chromosomes have moved in the middle. A spindle is formed. And in between the two centrioles, that equator of the cell, something like equator is formed. And the microtubules are attached to the spindles here. If you can see the threads of microtubules there. And this stage is referred to as metaphase. Next comes the anaphase. Anaphase means split. Ana means A-N-A, ana. Ana stands for splitting. So here what's happening is there is splitting up of chromosomes. The chromosomes undergo splitting at this stage. And at this stage, what happens? The cell can be differentiated into 46 pairs of chromosomes. So initially we have got 23 pairs. Now we have got 43 pairs of chromosome. After that starts the telophase. In the telophase, what happens? The two daughter nuclei are formed separate. So two nuclei are getting separate like this in telophase and division of cytoplasm occurs. The chromosomes elongate, they become distinct, nucleoli appear. The centriole of division, that is from here a constriction appears and divides cytoplasm, and thus two daughter cells are formed. This is all about the mitosis. In case of meiosis, we have got prophase 1, metaphase 1, anaphase 1 and telophase 1. So this daughter cell which has undergone mitosis, now it will undergo meiosis, M-E-O-I-O-S-I-S, meiosis. Meiosis is again divided into prophase, the first meiotic division. Meiosis is again divided into meiosis 1 and meiosis 2. So accordingly we have got prophase 1 and prophase 2. In case of prophase 1, what happening is the first meiotic division is very much prolonged, and it usually divided into number of stages like this. The prophase 1 again divided into the prophase 1 is divided into the following stages. We have got leptotene, we have got zygotene, the pachytene, and the last phase is the diplotene. So prophase 1 is divided into leptotene, zygotene, pachytene and diplotene. What is leptotene? Here what happens is the chromosomes are becoming much visible as in case of mitosis. The chromosomes are consisting of two chromatids. The chromosomes are consisting of two chromatids, and they are becoming more and more. Leptos means they are becoming more and more dense and distinct. Then zygotene. Zygos means what? Zygos means division, is it not? So the 46 pair of chromosomes, the 46 chromosomes, not pair, the 46 chromosomes in each cell, that is 23 pairs. The two chromosomes of each pair come to lie parallel to each other. So zygos means what? Zygos means doubling up or division. Division is zygos. So here two chromosomes, they come together and they pair, and this is called as zygotene. Then next comes the pachytene. The two chromatids of each chromosome become distinct and they form a tetrad. You are calling it as a pachytene. So they form a tetrad like this. They form a tetrad like this. So you are calling it as centromeres will come closer to each other and they form a tetrad. You are calling it as a pachytene. You are calling it as a pachytene. And next comes the diplotene. Diplotene, what happens is the two chromosomes of a bivalent now try to move apart, and there are breakpoints which are occurring here, and these breakpoints, something like this. And they start pairing with each other like this. So they start exchanging the material, and after that what happens is they become something like this. So are you able to understand the, so here exchange of material has occurred in between these two sister chromatids or daughter chromatids. So the two central chromatids become coiled to each other, they intermingle with each other, and they cross at certain places. I'm showing only one place of crossing here, but they cross at multiple places. I cannot show it owing to the reason that I'm unable to, I'll be confusing you rather than making you understand. So at different points, I'm showing only one point, but it occurs in different points. Breaking occurs and union occurs, and then a chromosome starts exchanging the material. So this is all about the prophase 1. Then comes the metaphase 1. Metaphase 1, again, the tetrads, they're lining up in the center like this. they're lining up in the center like this, and they form a line in the center, and thus there is division that is beginning. Then anaphase, what happens is the microtubule assembly starts pulling these apart, these tetrads apart, and at the telophase what happens is two daughter cells are formed with n number of chromosomes. If it is 2n, it has become n now, n number of chromosomes, n number of chromosomes. So then what happens is the meiosis 2 starts. Meiosis 2 again has a prophase 2, metaphase 2, anaphase 2 and telophase 2. Meiosis 2 is same like mitosis. So here there is no, what is the similarity of meiosis 2 and mitosis, they'll ask you. In case of meiosis 2, the number of chromosomes starting from prophase 2 are same as the number of chromosome in the daughter cells. So there is no half, unlike meiosis 1. In meiosis 1, from 2n number of chromosome it has become n, but here meiosis the daughter cells will have equal number of chromosomes, so it is more like a extension of mitosis. The meiosis 2 is actually extension of mitosis, right? So let us try to understand that this meiosis and mitosis are two different forms of cell division in case of molecular biology. Now at this point of time, I would like to show you one important diagram from Robin's textbook of pathology, and this diagram helps us to understand chromosome duplication, restriction points, and growth stages of the cell. Here we call it G0 phase, that is the resting phase of the cell. We call it G0 phase. From G0 the cell enters into G1, that is growth 1. Then we have got synthesis of DNA material. The cell is preparing to undergo division. Then we have got G2 phase where actual division starts, mitosis occurs, then after that immediately following G2 the actual division starts and mitosis occurs and cell division occurs. So we call it this as a cell cycle, and at this cell cycle we have got G1S, G2M. So in this G1, from G1 it goes to S phase, from S phase it goes to G2 phase, from G2 phase it goes to M, then division occurs. So here we have got two important checkpoints. There is a one checkpoint for DNA damage, we call it G1S checkpoint, and we have got second checkpoint is G2M checkpoint. At these two checkpoints, if there is abnormality, then the person may develop genetic disorders related to chromosome damage, and this results in serious genetic disorders. All right, so this is the cell cycle. We have got permanent cells which are like neurons and all, which do cardiac cells and brain cells which do not undergo this cell cycle much, but certain cells like hepatocytes, which are sleeping stage, they are not resting stage, we are calling G0 phase. From G0 phase they enter into G1, then S, then G2 and M, G1S, G2M, like that. All right, so to summarize we have got an interphase where DNA is replicated and preparation of cell division occurs. Then we have got prophase. In prophase, what happens to chromatin? The chromatin is becoming more dense. We call it condensation of chromatid. Condensation of chromatid is seen in prophase. Then we have got metaphase. In the metaphase, we have got the cells are lining up in the center and dividing at the level of equatorial plane. We call it metaphase. The chromosomes, not cells. The chromosomes are getting aligned like that. Then after that we have got anaphase where division occurs. The division is occurring like this, and then daughter chromosomes are formed. Then daughter chromosome gets separated in case of the telophase, and two separate cells are formed. This is all about the prophase, metaphase, anaphase and telophase. If you see meiosis is same like mitosis. In meiosis we have got prophase 2, metaphase 2, anaphase 2, telophase 2. In case of prophase 2, there is no DNA replication. Please remember this point. There is no DNA replication. Whereas in prophase 1, there was DNA replication. Then metaphase 2, same, the chromatids will come in the equatorial plane, and after that division occurs in anaphase 2 and 4 daughter cells are formed. All right. So a cell dividing into two daughter cells with same set of chromosomes. Say suppose 4 chromosomes here. So here we have got 4 chromosomes and 4 chromosomes. This is called as mitosis. Mitosis. Then in the meiosis, what happens is 2 daughter cells are formed with half the chromosome. Each of these daughter cells undergo division to form 2 more daughter cells, and this is meiosis. So we have got, this is meiosis 1 and this is meiosis 2. Combined together we are calling it as meiosis. All right. So this is the summary of today's class. We have understood genetics and molecular biology. All right. Hope you have understood the class, and any further doubts, please definitely ask me. I'll be very, very happy to help you out in solving your doubts, right? Thank you so much for patient listening. Bye.